Hey everyone, welcome back to Windows Seed Studies. I hope you all are doing well. So in last class we were learning about non-coding RNA. Today we are going to learn about long non-coding RNA. So those are the RNA which do not code for any protein. So the long non-coding RNA being long, they are more than 200 nucleotides long. Okay, but there are a certain characteristics of long non-coding RNAs, like they are processed just like mRNA, the messenger RNA. So means they can be spliced, they can be capped, they can be polyadenylated. So the processing happens in the exact way as of the messenger RNA. The other characteristics of long non-coding RNA is that they are constrained to the nucleus. So all of the functioning that they have to do, that happens in the nucleus. Okay, so we are going to learn in detail how long non-coding RNA they are able to regulate the epigenetic mechanisms. Okay, so we are going to take a look at epigenetic regulation in detail of the long non-coding RNA. Okay, so this is the goal of the lecture. Okay, first thing that when we were discussing about chromatin remodeling complexes, we discussed that those are the complexes which change the chromatin environment. We discussed about SWE-SNF complex. I hope you remember that. So when we discussed about those, we got to know that those are recruited to the DNA by some site-specific binding proteins or the sequence-specific binding proteins. Okay, so we learned that chromatin remodeling complexes, chromatin remodelers, they are recruited to DNA with the help of sequence specific or site specific DNA binding proteins or the proteins. Okay, it's not necessary that they go and bind to the DNA. So this is what we discussed. But the question was how those sequence specific proteins are able to bind to the DNA, because that DNA is already occluded by nucleosomes. So that DNA is inaccessible to any reading machinery possible out there. Like it can't be read by any kind of enzymes, any kind of transcription factors. But then we got to know that there are certain exceptions like pioneer transcription factors which can recognize the nucleosomal DNA as well. But alongside that, there were other mechanisms that can be adapted for the recruitment of the chromatin remodelers. So those were, for example, the nucleosomal breathing. So nucleosomal breathing is transient unwrapping of the nucleosomal DNA, which makes the DNA, which was previously occluded by nucleosome, now accessible. So now it is accessible and it can be read by the transcription factors or that DNA binding proteins, and those DNA binding proteins recruit the chromatin remodeling complexes. The one specific mechanism that we discussed at that lecture also was that those chromatin remodeling complexes that can be recruited by the non-coding RNA. So here the non-coding RNA helps, okay, and that the non-coding RNA that helps is long non-coding RNA. Okay, so we are going to take a look at the mechanisms by which lncRNA is able to recruit chromatin remodeling factors or any other factors which are going to change the chromatin environment, and how this results in the epigenetic regulation. So for regulating the transcription, you can change the chromatin state. You can make the chromatin, you can make the chromatin more dense, or you can loosen up the chromatin, and this changes the transcription ability. Means it can either activate the gene or it can repress the gene. So for changing the chromatin environment, you need to recruit certain factors, or you can do the modifications on the histone or on the DNA. So the modification part is covalent part, but how do we change the chromatin environment non-covalently? That is done by the chromatin remodeling factors. And for the recruitment of chromatin remodeling factors, long non-coding RNA are going to help. And this is how they control the epigenetic regulation. So like this is how they change the chromatin environment by recruiting the chromatin remodeling factors. So now the question is how the long non-coding RNA can access the DNA which cannot be accessed by certain other DNA binding proteins. So this is due to the characteristics of long non-coding RNA. First, its length. Okay, it has certain length, like it is more than 200 nucleotide long, and again it has a sequence. Okay, so the sequence of long non-coding RNA makes it specific for certain sequence on the DNA. So like the complementarity of the long non-coding RNA to the DNA sequence makes it very specific. So the length and the sequence of long non-coding RNA ensures the specificity of the long non-coding RNA. The specificity here means that long non-coding RNA will go and bind to the certain sequence there where it should be. Okay, so like depending on the need, it will go and bind. So we are going to take a look at the mechanisms by which it does it. It does its job. The first mechanism is guide. Okay. Second is scaffold, and the third one is decoy. Okay, so that those are the different mechanisms by which the RNA works. So in the guide, as the name suggests, in this the long non-coding RNA interacts with the chromatin remodeling complexes and it acts as a guide. So for example, this is a factor. Okay, so this is a certain factor which needs to be recruited on this DNA. Okay, so now the non-coding RNA. So this is the non-coding RNA. Okay, lncRNA. It will bind to this chromatin remodeling factor and it will act as a guide. So due to its sequence specificity, it will go and bind to the certain DNA sequence and it will act as a guide for this factor. So the long non-coding RNA is already bound to the factor, the chromatin remodeling factor. Now it will act as a guide and it will recruit that factor to the DNA. So now the remodeling factor can do its job. This is how the guide mechanism work. But the scaffold mechanism is a bit different. So in the scaffold mechanism, the long non-coding RNA, it serves as a central platform to which multiple complexes can come and bind and they can do their function. So what happens is that long non-coding RNA, this works as a central platform. Okay. So for example, now this is the long non-coding RNA. Okay. And to this, multiple factors can come and bind. And now those binding of the multiple factors to, binding of the long non-coding RNA to DNA, this provides a platform for all of those complex to come, sit there and work. So they can transiently assemble due to the help of long non-coding RNA to the place where they need to do their function. And when they assemble there, they do their function and then disassemble again. So transiently they can transiently assemble on the central platform provided by the long non-coding RNA and do their work, do their job, and then can disassemble. Okay, so like long non-coding RNA provides a dynamic scaffold which can change now the chromatin environment. The decoy mechanism is quite distinct because here it doesn't act as any kind of platform or it is not guiding any transcription factor or any other chromatin factor to the DNA. But here the long non-coding RNA itself goes and it binds to the transcription factors which are required for the activation of gene, or it binds to the microRNA. Okay, so we know that microRNA does the post-transcriptional silencing. So binding to the transcription factors or microRNA inhibits their binding to the DNA sequence. Okay, so though like they are held captive by the long non-coding RNA. So long non-coding RNA, when it binds to the transcription factor, it inhibits the activation of gene, or when it binds to the microRNA, it inhibits the mRNA degradation, or it inhibits the silencing of that particular gene. So this is how the different mechanisms of the different mechanisms by which long non-coding RNA works. Okay, and let's now take a look at the examples of long non-coding RNA. Okay, before discussing about examples, let's take a look at the categorization. So those long non-coding RNA, they are categorized in different uh, types depending on from where they are transcribed. The first one is antisense lncRNA. The other is exonic lncRNA, intergenic lncRNA, intronic lncRNA, sense lncRNA. So there are multiple types depending on the from where those are transcribed. So antisense long non-coding RNA means those are transcribed from the antisense strand of the gene. For example, this is the gene. Okay, the DNA sequence. Let's say that this is the DNA sequence. This is the DNA sequence, and this part is a gene. Okay, so now this is the double-stranded DNA, right? And when this part is transcribed, we this part is gene. This is the sense strand of the gene, and this is the antisense strand of the gene. Okay, and when the antisense strand means the strand which is opposite to the strand which is coding one. Like the strand which codes for the gene is known as sense strand, means which makes the protein when upon getting translated. But the strand complementary to it is known as the antisense strand. This is the opposite strand. Okay, so this antisense strand, when it is transcribed, it makes a long non if it makes a long non-coding RNA, that kind of long non-coding RNA is known as antisense long non-coding RNA. Those examples, exonic long non-coding RNA, so those are those are transcribed from exons. The intergenic long non-coding RNA, those are also known as link RNA. Okay, so those are transcribed from the regions between the genes. So between the genes, there is long non-coding region. Okay, this does not code for any protein, but they can code for the lncRNA. Okay, when they transcribe, they can form the long non-coding RNA. So such long non-coding RNA are known as intergenic long non-coding RNA. If the intron lariates, intron lariates, I hope you know in the splicing, the intron lariates can form the long non-coding RNA. So such are known as intronic long non-coding RNA. And if the sense strand makes the long non-coding RNA, those are known as sense lncRNA. So those are the different types of long non-coding RNA, which those are categorized based. The categorization is based on the transcription site, like from where the lncRNA is transcribed. Okay, from depending on that, those are the different categories. And depending on the functioning, there are multiple categories. Like for example, the long non-coding RNA, which are involved in X chromosome inactivation. Okay, we will learn in detail about all of those in upcoming lectures. The long non-coding RNA which are involved in X chromosome inactivation are exist, as well as T6. Okay, those are both of those are long non-coding RNA. There are long non-coding RNA which are involved in genomic imprinting. Okay, so the examples are KCNA, NQ1OT1. We will also discuss about this in detail. There is AIRN, long non-coding RNA. Again, there are long non-coding RNA which are involved in other mechanisms, like for example, Hox gene silencing, the hot air one. Okay, we will learn about those lncRNA in detail. How do they function and what do they do in detail in the next lecture. So this is all about the long non-coding RNA. We learned about the mechanisms by which they work. Like for they can work as guide, as a scaffold, as a decoy. But these are the main mechanisms by which they function. Like these are the main. But alongside those, there are other mechanisms. Okay, for example, when antisense long non-coding RNA, okay, this antisense one, antisense, it can work through other mechanisms than guide, scaffold, or decoy. For example, it can do the transcriptional interference. Okay, let me brief this those mechanisms once. Transcriptional interference means it can physically interfere with the transcriptional machinery, means it can bind to the transcription factors which are important, like we learned in the decoy mechanism, right? In the decoy, it binds to the transcription factor, but it can also physically collide with the transcription factors or the factors which are required in the transcription. So it can interfere with the transcriptional machinery, or it can form the RNA DNA hybrid. Okay. Okay, so there's a 3D structure which is known as R loop. Okay, if it is formed, the transcription is hindered. So the formation of RNA DNA hybrid is possible due to long non-coding RNA because the antisense strand has the complementarity with the sense strand, right? So that is why the long non-coding RNA can go and bind to the and can form the R loop. It can form the RNA DNA hybrid, which will inhibit or which will interfere with the transcription. Again, there's a possibility that it can lead to the either mRNA degradation, or it can lead to more stabilized mRNA. Okay, so it increase or decrease the mRNA stability by binding to the mRNA. If it decreases the mRNA stability, or if it increases the mRNA stability, it will directly result into the elevation of transcription or the suppression or the inactivation of the gene. So these are the other mechanisms, the mechanisms other than guide, scaffold, or decoy, by which those long non-coding RNA can work. So, but those are very common mechanisms, but these were quite different ones. That's why we discussed those all in detail. So this is it for long non-coding RNA. In next lecture, we will learn about the exist, the hot air RNA in detail. We will also discuss about X chromosome inactivation, genomic imprinting, dosage compensation, all of those in detail in next lectures. Till then, see you. Bye bye.